Okay, let's continue. In the This is our reverse, forward and reverse. These are our forward and reverse strands. We've selected both of them, just like I explained. You hold down your control key, select forward, then select reverse. This, remember, this is the aligned sequences. Remember, when you're working with a particular phase, like this is interface, make sure it's selected. That's what you're working on. So we it's highlighted. So we go to align and create consensus sequence. Once you click on it, you see another result comes up as consensus. See, look at the consensus sequence. So look at the consensus sequence. We can decide to copy out from, this is an example that we are using, so it may not be so perfect, but in the real sense of it, it will almost be in the consensus sequence. If you look at it, we are seeing gaps. Okay, these gaps you need to break it out and fill them up. So what do? Because it's highlighted, we copy the uh, consensus sequence. Go to we copy so we copy it in a faster format. So if you go to edit, you can copy. There are different copy sequences, copy sequence vertically, copy sequence to clipboard in a faster format. If we copy a consensus sequence and we go to notepad, any anyone, we copy, we paste it. So look at our sequence. So you see, for us to use this sequence, we just remove this, remove this, we remove the gaps. This is an example in a more ideal sequence. It won't have much gap. It won't have much gap. So I may not remove all the gaps, but it's ideal to remove the gaps. But we can continue from this here. So assuming this is our consensus sequence, so this is what we are going to work with. This is what we are going to use to query the database. How do we query the database? To use this sequence to identify our organism, identify the gene we are working with, find out other sequences that are closely related, and other things like that. So assuming we have finished and this is our sequence, let me call this consensus. Remember, it must be in a faster format. This is faster. Faster must have a greater than and an ID. So this is the faster. So if I copy this, Control C, go to NCB, you Google NCBI or NCBI.org, just NCBI, is the National Center for Biotechnology. It will open up. It will open up something like this, and you click as ncbi.nlm.nih.gov. This is their, the home page. NCBI. But if we are looking for blast, basic local alignments, search tool, we use it to search the database, existing, to compare our sequence with others and know which organism or which gene is our sequence related to give us some certain basic information. So if we click on this blast, it will open up blast, different blast, nucleotide, the one for protein and so on. But we're interested in nucleotide, so you click on this nucleotide blast. It will open up in this one. If you look at this blast, You can enter a lot of things. Enter your sequence here. If we have saved this sequence, maybe as a notepad, you put it in notepad, save it, you can upload it here. If we already have maybe an accession number, you can enter session number. But we remember what we have is the faster sequence. So I can create a faster sequence here. And I will suggest you can leave now, leave other parameters, standard database, and go down and blast. Remember, we're blasting our consensus sequence, and they will tell you the job is running. The job will be running on take 3 seconds, depending on your browser and your connection. So let's just wait 1 minute. Start it running. Your job title. Remember that's one thing. Okay. So look at this result. This is the result, the last result. So if you look at it, you see from beginning. You see the job title consensus blast N nucleotide blast means because nucleotide that we're looking at. Remember that are protein. But now here we're looking at nucleotides, and the molecule type is DNA. The query length is 1154. That means our sequence is 1154. So always note the query length. So if you look at it, you see description. There is graphic summary, there is alignment, there is taxonomy, but let's focus on the description. So if you look at the description, you see the first that comes up is Bacillus species, Bacillus cereus strain, Bacillus cereus strain, blah blah blah, Bacillus cereus strain, ribosoma 16S. Actually, our sequence is a 16S ribosoma RNA gene from an organism, but we are not sure of the organism. So this is it. We are trying to identify the organism. So if you look at this first hit, if you look at all of them, they are like bacillus. So this can tell us that our organism is bacillus. In the first, you'll be seeing Bacillus species, second cereus tropicus, atracis. They're too sure. So this is an already existing strain. Actually, that's what I used. We studied earlier as NADU. We studied as Bacillus species because very sure about this. It wasn't so specific. At times 16S may not be so specific, so but it can give you that it's a particular organism, but the certain strain is may not give you a certain time. So but sure it was Bacillus, so we studied at Bacillus species. You see that what we studied as 100%. That's the only 100%. It means it has already been studied. So we use an example of the one we have worked on before and we studied. So the thing when you get to the right part, you see this is some kind of focus. So the query cover, I did not use the consensus sequence. I just used one sequence just for example purpose. So query cover, you must know the query cover, and you must also know the percentage identity. E value is like the probability value. The lower it is, maybe 0.0, 0.01, the lower it is, the more accurate. So the query cover means that our sequence covered with the one it's comparing to about 51%, 88%, and it's identical by 100%. It's a very good one. So I don't want to go into percentage identity, percentage similarity, but here percentage identity is what is referred. So normally you select the first hit. So if we click on the first hit, it will take us to the alignment. Look at the first hit. This is our query. This is our subject. So you see the first hit, Bacillus species, 597 16S. Okay, now look at this. Query is 130. Subject. Okay, look at this. This query subject, query subject. You know, it's comparing our query with another one closely related in the database. So from our query from 130 base to 189 is similar to a subject in the database from 600 to 541. So that's how it's 189, 190, 541, 540. So you keep comparing them, aligning them, comparing them bit by the sequence, session by sessions, until down to this place. So we can look at the gene bank. Very important place to look at the gene bank of this particular gene, bacillus species. If you look at the gene bank, you have information about the gene. So if we click gene bank of this species, so I'll show you something. This gene bank is how it gives information about the particular organism, where it was selected, where it was, how it was sequenced, and all the information. See the gene bank, it has a session number. If I carry this accession number, I copy it and go to NCBI database, the web page. If I want to go straight to this gene bank, I'll select nucleotide here under our database. I'll select where is nucleotide, and if I paste this accession number here, if I click this, it will take me directly to the gene bank. Same place. Same place. Directly to the gene bank. So in the gene bank of this, what information can we get from here? We can say that, oh, our accession number of the gene. No, I told you when you sequence, you can store it. So you see, locus is this of the gene, 600 base pairs. The gene is 600 base pairs sequence, and it's linear DNA. Bacillus species. If you go down, there are authors. In some cases, the papers are published, but if this is not published, look at comments, assembly data, sequence technology, Sanger sequencing, what was used. So you can see all that information, sources from 1 to 600, 1 to 600. You can see the collected by the name of the person that collected it, collection date is in this country, this host, this organism, Achatina. The isolation source from a forest. So you see, collection date was in April 2017, but this gene was deposited in February 2019, 22 February 2019. You can see that. So these are the same gene. Okay. So this information is very, very important. You can, find out. You can have your session number. If you have your sequence, you can deposit. In one of the videos, we describe how you deposit your sequence to NCBI database, DBPJ database, whatever. It can be a protein sequence, it can be a protein sequence. So when you deposit it, you get your own accession number. So in the next video, we'll describe how to get our what is it called, build our own phylogenetic tree. Build our own phylogenetic tree in the next video. Thank you.